本次会议的第2场专题演讲即将开始，欢迎主持人陈勇志教授。The second invited speech will begin. Please welcome our moderator, Dr. 陈勇志。
Hello everyone. It's very good to see so many people and also friends from the other side of the world to join this conference and also keynote speech. Today, I'm Chen Yongzhi from National Taiwan Normal University, Department of Sports, Physical Education and Sports Sciences. I'm also a moderator for this keynote speech. And allow me to roughly introduce our keynote speaker, Dr. Tatsuro Amano. His research is focused on the sweating physiology and thermal regulation, and as well as heat adaptation. And he has also published over 100 research papers in very good journals, such as Journal of Applied Physiology, and American Journal of Physiology, and also European Journal of Applied Physiology. So they are all very good quality journals. So you can see his work is very high quality and also very productive. At the moment, he's also leading a group of the undergraduate and postgraduate students in the laboratory for exercise and environment physiology. Okay, so today his keynote speech will be about the non-invasive way to evaluate sweat electrolytes and lactate. So let's welcome our keynote speaker, Dr. Amano.
Hello everyone, and thank you very much for your introduction. So first of all, I'd like to thank the conference organizer for inviting me to this conference. And this is my first visit to Taiwan, and it's hot, but here it's cool, so it's okay. So I think, because I'm not very famous researcher, it's good to introduce myself first. So I come here from Japan, and Niigata is located around here. So this is my lab members. Now I have in total 9 members in the lab, and 2 are from China. So I'm happy to work with East Asian countries. So Niigata is relatively north in Japan, so we have snow in winter period. And I'm interested in local mechanisms of sweating and cutaneous vascular responses. And the laboratory mainly focused on these 4 primary topics, including whole body human thermoregulation, in vivo sweat gland physiology using the pharmacological approach. And that's why I'm interested in transdermal drug delivery techniques to develop the use of human sweating research. And we also do some kind of hydration studies, developing liverage using some kind of ingredient. But today I'm talking about the sweating content. So this is what I will talk today. So firstly, I'm going to introduce about basic physiology of human sweating. Because I don't think here not so many people know a lot about sweating in humans. And then I'm going to summarize about the variations of sweat rate, sweat sodium, and lactate concentrations. So especially in terms of methodology and physiological interpretation. And then finally, I will introduce recent our work regarding this topic. And honestly, we have just started about the research of sweat content during the last 2 or 3 years. So the research is still running. So maybe I cannot show kind of a complete result, but I'm happy to share some of our work. Okay. So during the exercise, we know that heat is generated in the body, especially the muscle. And this heat production is quite huge. And this is just an example. When one runner body weight is 78 kg with weight mass of like 60 milliliter per kilogram per minute, and when he runs for 60 minutes at middle exercise intensity, heat can be generated around 1000 watts in the body. And when considering the heat content, this amount of heat is corresponding to core temperature elevation of 13 degrees Celsius when no heat loss happens from the body. And I think you can imagine this never happened because our body cannot maintain the life in this situation. And this is the actual result reported by their study. So actual core body temperature elevation, so this is SYU temperature, is approximately 1 degree Celsius from baseline. So most of the heat is likely dissipated from the body. So when the heat is generated in muscle, the heat is circulating by blood flow, and then core temperature elevates. So this also activates to the thermoregulatory center of hypothalamus to induce sweating and cutaneous vasodilation. So this is a study reported by Glen Kenny Laboratory from Canada. So which shows the rate of heat loss by sweat evaporation and dry heat loss from the skin. So as you can see, especially when sweat is evaporated almost free, the rate of heat loss is mostly from sweat evaporations. So sweat is important for maintaining exercise in hot function. This is another example because I work in physical education or faculty. I often talk, not often, maybe twice a year, I have to talk in front of elementary school students. So then I use this one. So we can visually know that before exercise, skin temperature is high, but during running in hot conditions, skin temperature gradually decreasing because of sweat. The runner is sweating. Finally, the skin temperature is quite lower than baseline levels. So this is also introduced in language textbook in Japan in elementary school. So I wrote some kind of sentence for elementary school titled low sweating or something like that. Okay. Then, so I want to go a bit more deep in sweat gland physiology. So sweat glands locate approximately 1 to 3 millimeters beneath the skin, and there are almost 2 million functionally activated sweat glands in humans. So as you can see, the sweat gland is mainly two portions. So here is secretory portion to produce sweat, and then this is the distal duct portion to kind of way which appears sweat on the skin. So this is another illustration which is more easy to understand. So this is secretory portion, so secretory cells, and then this is ductal position. So when sweat is generated in secretory region, the initial sweat contains high sweat sodium concentration, which is similar to blood sodium concentration. But when the sweat is running through the duct, the sweat sodium is reabsorbed to the body, and which appearing hypersonic sweat on the skin. So that's why sweat is some kind of two steps. So generating and reabsorbing. So this suggests that sodium reabsorption capacity of sweat glands may determine sweat sodium concentration. So this is more mechanistic illustrations. So this is a little bit confusing. So in summary, what is important here is that when neurotransmitter acetylcholine is released from the nerves, muscarinic receptor is activated, and then calcium concentration increase in the cell. So this elevation induces several movement of ions like potassium, chloride, and sodium. So what is important here is that finally chloride ions is accumulated this side. So this is the dark side, and this accumulation generates chemical gradient from basolateral membrane to here, which encourages the movement of sodium from here to here with water. And then when sodium moving to the dark space, the sodium is reabsorbed to the body through sodium channel and sodium pumps. And in terms of sweat lactate, when you look here, there are several this is actually sodium pumps which use ATP to maintain the sodium balance in the cell. So because sweat gland also needs ATP, there are some aerobic and anaerobic metabolism in the cell. And sweat lactate is a byproduct of glycolysis in sweat glands, which is generated in sweat gland cell. So this sweat lactate is moved to sweat this side, although the precise pathway is still unclear. And this is the reason why sweat contains amount of lactate. So this suggests that ATP supply around here to produce sweat may determine the sweat lactate concentration. So when we look at regional variation, there is a huge variation in sweat. So this is kind of famous article reported by Radcliffe University in the UK. So it shows that during exercise, this is middle and high intensity exercise. We sweat mainly posterior side, higher sweat rate posterior side relative to anterior regions. And there are more sweat in the central region of the body relative to the extremity. So we also sweat head a lot forehead during exercise. So when we look at the number of activated sweat glands or sweat gland density, this is a little bit different. So I create this figure using ChatGPT based on these values. So as you can see, there are many sweat glands on arm and toes, and fewer glands exist in the main body. And more sweat glands exist at the head and looks like gradually decreasing to lower bodies. So as I said, there are more sweat back and also central side. But as you can see, the number of sweat glands doesn't explain the regional variation of sweat rate, indicating that sweat gland AC may more influence the sweat rate distribution during exercise. So this is the sodium concentration distribution. So this is also created by ChatGPT. I put the data reported here. So as you can see, the sodium concentration is likely relatively higher in upper bodies, and then reducing the lower bodies. However, it is also reported that sodium concentration can differ depend on the heat stress, gender, and any other confounding factors. So this is kind of average understanding. So there are more individual variations in sweat. So this is summary from US group, Gatorade group. So it is sweat rate and sweat sodium concentration. So this graph shows the frequency for how many number of athletes who sweat in liter per hour in this graph. So in average, of course, it depends on the exercise intensity. Athletes can sweat almost 1 liter per hour. And some can sweat more than 3 or 4 liters per hour, but myself, I haven't seen such a huge sweat. But it is reported sometimes. And sweat sodium concentration, the average concentration is like 40 to 50 millimole per liter. This is forearm, and this is whole body response. I will explain how to measure whole body sweat sodium concentration later. So, what is important here is there are huge individual variations in sweat production and sweat sodium concentration. And I don't know about lactate data similar to this. So then I want to introduce how to evaluate sweat rate and sweat sodium and lactate concentrations. Firstly, I'm going to introduce methodology. So sweat rate, it is often used to, which is called ventilated capsule method, to measure local sweat rate continuously. So this is probably the first research which reported how to measure local sweat rate by using this method. So here, they used compressed gas running through the tube, through the skin, and then the humidity of the gas is reduced, and then dry gas is diffusing to the skin. So this is kind of capsule. And then this dry gas includes some humidity which is measured by sensor to determine sweat rate. So basically this method is still used in many laboratories in the world. So there's still standard method to measure sweat rate continuously. This is a picture of myself. We attach the sweat capsule on the skin by using the collodion. Collodion is a kind of glue which is available for human skin. So this is skin and this is capsule. So dry gas, so we buy compressed dry gas, so already dry gas, packaging to the capsule to measure sweat rate. So this is actual data measured by like this, so similar place of the skin. So as you can see, the response is quite similar, synchronized in different parts of the body, because this is because sweating is regulated by autonomic nervous system. So the response is really similar for bodies. When we use atropine, this is a drug to inhibit sweat production. So you can see no sweat happen under the capsule. So this suggests that that's why this is kind of a difficult method to measure sweat rate. So this is good to measure continuous and precise measurement. But unfortunately, this method cannot correct sweat itself, so we cannot analyze sweat chemical composition by using this method. So that's why several laboratories use another method to correct local sweat, which is absorbent patch and cotton method is most often used for this purpose. This is quite simple method. So we apply cotton on the skin, and then paraffin is covered, and after that paraffin is covered, something like this. So sweat rate is determined by the weight changes before and after exercise, and sweat composition is analyzed by chemical analysis from the sweat from cotton. So, and also there is another method to correct sweat, which is called product sweat corrector. Unfortunately, in Japan we cannot use this because there is no way to import this. So maybe this is used in US, I think, because this is developed for the diagnosis of cystic fibrosis, and which is often used in clinical situations. So So when participants sweat, this tube contains sweat and then color changing, and which can determine sweat rate. And sweat content can be analyzed or directly collecting the sweat from the tubes. So these methods can analyze sweat rate and sweat compositions, but unfortunately it's not continuous measurement. And this method requires a certain amount of volume, so low level of sweating is a little bit difficult to detect. So this is kind of challenging for these methodologies. So that's why several researchers try to continuously assess index of sodium or electrolyte concentration. So this is a report from Japan. It's oral study. So they developed microfluidic disc which perfuse ultra pure water into the tube here, and this is the skin, and here is the perfusion channel. And they measure the conductance of sweat of the perfusion water by using the sensors. So this is changes in heart rate, sweat conductivity measured by this technique, and sweat rate. As you can see that 10 participants doing hand grip exercise, heart rate increases, and sweat rate measured by ventilated sweat factor, and also sweat conductivity increases with synchronized response. So this suggests that this method may be available for continuous assessment of index of sweat, sodium or electrolyte concentration. So this device is also used for heat stress test, which is conducted by my supervisor laboratory, although I was not there at that time. So they assessed index of sweat ion concentration measured by this technique and plotted the response, the relationship with changing sweat rate measured by ventilated sweat capture method. And then they found the non-linear response of this relationship, because classically it is believed that these relationships are linear, but it looks like there is a kind of threshold to increase the sodium or electrolyte concentration in increasing sweating rate. And then when I was a PhD student in my previous university, Kobe University, so we worked to try similar technique, but this is more simplified response. We measure skin conductance, just applying the conductance sensor on the skin, combined with sweat rate measurement. So skin conductance is known to general hydration status of the skin, so which may be similar to this assessment. So by using this method, we also found kind of a threshold response, which is more evident after heat acclimation protocol. So indicating that this method may be useful for detecting index of sodium concentrations. However, although these techniques are possible to assess low range of sweat sodium concentration or kind of index continuously, but it's not direct measure and absolute values are unknown. So this is a big problem in this method. So recently, this is not my work, but I'm working together with them in Japan. We developed lactate sensor. This is not sodium sensor. Lactate sensor to measure sweat lactate concentrations. So they developed a similar technique as I showed in previous slide, which shows the water is saturating to the skin, and then sweat lactate can be detected by the sensor, which is specific to the sweat lactate concentration. So what is important here is that this sensor can measure sweat lactate concentration and also we can determine the sweat lactate content in sweat, the absolute amount of sweat lactate. So this is quite important for physiological studies. So I will explain later why the reason. So this looks like a very good device. However, human data is quite limited. So they reported that just only one participant exercise, intermittent exercise, they assess sweat rate and also sweat lactate content by using this device. So they say it looks like synchronizing sweat rate and sweat lactate content. So it looks okay, but it's not scientifically valid. So actually more human data is required by using these techniques. So although I don't work a lot about this area, there are so many developments to measure sweat content, sodium or metabolites recent years. So it looks like, I think the common way method to measure sweat component is combination of calorimetric technique and also the microfluidic techniques. So I cannot follow all the papers because there are so many engineering papers reported to measure sweat content. But I think still it's challenging to continuously measure sweat rates with low to large high level of sweating. And also these engineering studies are a little bit enough for better physiological interpretations. So they can measure, but they just measure. So it's quite difficult sometimes to interpret the result. So that's why it's important to know how to interpret the results of sweat sodium and lactate concentration. So I will introduce about how to interpret physiologically. So this is also practical data from US. Professor Bono's laboratory. So this is simply shows that when sweat is collected by absorbent patch method and plotted with the relationship, and then it looks like this relationship is quite linear. This is okay. And what is important here is that they calculated sodium reabsorption rate by using this formula. So they also measured blood sodium concentration to determine the sodium content. And they showed that sodium separation rate and reabsorption rate both linearly increase in response to increasing sweating rate. And this difference is considered sweat sodium content. So these reports show that sweat gland sodium reabsorption capacity represented by the slope over here may determine sweat sodium concentration. So this is another report from the same group. What is important to measure sweat sodium concentration is that if we measure sweat sodium concentration alone, for example, when we obtain the data value like 50 mmol per liter, it is a little bit difficult to interpret because, for example, before an heat accumulation, it is known that sweat sodium and platelet concentration shift to the rightward, like here. And then we measure only sweat sodium concentration, and if we don't know the sweat rate, and if one participant's sweat very low but recorded this value, I think this may be interpreted as salty sweat because it is relatively concentrated at low level of sweating. On the other hand, after heat accumulation, thus even if we obtain the same value, but if he or she sweats more, it may be considered well regulated because this participant doesn't lose a lot of sweat despite they sweat a lot. So that's why sweat sodium concentration alone can induce misinterpretation in physiological response. But however, there are many methodological challenges for better physiological interpretation of sodium concentration. One is that this graph again looks like looking at the relationship with sodium concentration, sweat rate. This graph looks okay, but this is mean value, mean response. I think when we actually collect sweat from absorbent patch method, it's quite difficult to collect sweat something like this. So some patches can be something like this, which is not linear, and some others can be like this, so which lacks middle level of sweating. So this suggests that the true relationship between sodium concentration and sweat rate is a little bit unclear, although mean average response looks like linear. So this is this is data from myself, so index of sweat electrolyte concentration, so in conductance response. So as you can see, some changes can occur at the low level of sweating, like something like 0.2 here, and 0.2 is here. So as I said, low level of sweating is a little bit difficult for challenging for absorbent patch method. That's why obtaining this type of data for all participants is a little bit difficult. Another consideration of this technique is that they usually sweat during exercise or heat stress, but these interventions can induce a change in skin or temperature, and also it can induce dehydration, which also affects the sodium concentration. So in terms of physiological interpretations, it's better to look the local effect, so how locally sweat gland is modulating the sodium concentration. So continuous assessment of sweat sodium concentrations with standardized sweat production to cover low to high sweat rate without exercise or heat stress may be ideal for better understanding of physiological response in sweat sodium regulation. This is maybe related to pharmacological interventions. Then when we look at sweat lactate content, this is relationship with lactate and sweat rate. So I think we can expect that sweat lactate concentration also increase with increasing sweating rate, because sweat lactate is produced based on sweat gland activity, so ATP requirements. However, it's interesting that sweat lactate is a little bit different. So it is known that sweat lactate concentration decreases with increasing sweating rate. This is because which is called dilution effect. So because when sweat rate increases, sweat lactate also increases, but relatively sweat production is larger than increasing sweat rate, reducing the concentration of sweat lactate. And it is also known that after heat accumulation, so this relationship can be changed, something like this. So after heat accumulation, sweat lactate is less produced in response to the level of sweat production. Actually, the reason this reduction is not clear, but may be affected by the metabolic oxidation, so sweat gland activity can be more aerobic after heat accumulation. So, like a sweat sodium concentration, it looks like sweat lactate concentration also need careful interpretation when we measure this parameter. So they also reported individual variation of this relationship. So as I said in previous slide, individual response is quite different. So some people I think not many participants can sweat from low to very high level of sweating. So just only high level or middle. So the relationship is quite varied individually. So as I said, clear relationship is not clear. So the challenge is the same. So lack of sweat rate, low sweat production, lack of wide range of assessment, and that's quite a precise relationship actually. So I think one way to induce sweat is another is pharmacological interventions. So this is my favorite method to induce sweating, or to administer any kind of drug to the skin, which is called anti-inflammatory. So to induce sweating, some kind of drugs such as alkaline, this is agonist to muscarinic receptors in the sweat glands. So we apply small current on the skin, which enhance the transdermal delivery of the drug to the skin. So some research use this technique to assess sweat sodium concentrations. For example, this research collected sweat using macroduct sweat collector. And then compared sweat sodium concentration across athletes. So they reported higher sweat sodium concentration in football, baseball, and basketball players compared with soccer and rugby. But this is a little bit quite difficult to interpret because they just measure sweat sodium concentration and it's not clear physiologically because although they collect sweat the same amount, but they don't adjust the collection time. So this is quite important because when sweat is produced here, the reabsorption rate is influenced by the speed of passing sweat to the dust. So slower movement can induce the efficient reabsorption, but higher speedy passing is less efficient. So that's why sweating collection time is important. So, okay, then I summarize the challenge in methodology and physiology. This is kind of a summary. I need to rush a little bit. So based on these summaries, we have recently conducted some studies for better understanding of physiological interpretations. So firstly, we did pharmacological evaluation of peripheral sweat gland regulatory capacity. So what we did, we recruited trained and untrained participants, and we conducted alkaline anaphylaxis to induce sweating. So after anaphylaxis, sweat rate increased, initially very high level, and then gradually decreasing over 2 or 3 hours near to the nearing zero. So we collect sweat by using absorbent patch method, and then we created sweat sodium concentration and sweat rate relationship from very high to very low levels. And we determined the slope of this relationship. So this is the data, sweat rate and sweat sodium concentration. So trained participants sweat more during the initial phase compared with untrained participants. But sweat sodium concentration looks similar. So we created this relationship using individual data and determined the slope of the response. So as you can see, the trained participants' slope is less than the untrained participants. So this is statistically different. So this indicates that trained individuals can maintain the sodium concentration despite the high sweat rate. So this is a kind of training-induced adaptation. So we also explored another way, because in this experiment, it takes almost 3 hours to collect all the samples. It's a bit time wasting. So we analyze the initial data only. This can work to reflect the sweat gland capacity to regulate sweat sodium. So we found the similarity of this response compared to the slope obtained from the whole data. It looks that 15 minutes analysis is still okay to determine the sweat capacity of sweat gland. And we also explored whether the sweat gland sodium regulation capacity contributes to individual variation in whole body sweat production. So this is a brief background. So recently it is reported that whole body sweat sodium concentration is mostly influenced by the energy expenditure. So this is exercise intensity. Maybe because of sweat rate is increasing depending on the exercise intensity. However, this is not really physiological because it's not surprising that sweat rate is independent of exercise intensity and therefore the sweat sodium concentration. So physiological basis of this response is not clear and should be related to sweat gland capacity itself. So to determine the physiological contribution, we employed a protocol to control sweat production because sweat rate is always compounding factor to determine the sodium concentration. So we did whole body washdown technique to determine whole body sweat composition. So firstly, participants were cleaned by ultra pure water and then exercise for 90 minutes in hot conditions. And the exercise intensity was determined to evaporate the requirement for 200 watt per square meter. So this is a technique to minimize variation in sweat rate. And then the participants were washed again to determine whole body sweat sodium loss. So we compare these responses against the local method. So this is the relationship between whole body sweat sodium concentration and whole body sweat rate and energy expenditure. So we cannot see any relationship. So this means that we could control sweat production. So when we put whole body sweat sodium concentration to the response of peripheral regulatory index, we can see very weak but statistically significant relationship. So this may indicate that peripheral sweat gland capacity weakly or partially explains the variation of whole body response when sweat rate variation is minimum. And similar result was obtained in the 15 minutes response here. So also indicating this is also a good technique way to evaluate the sodium concentration. Okay, so finally we try to assess the lactate content continuously. So we developed another device as I have explained previously. So EBS is fused into this device and we determine lactate content continuously. So sweat rate induced by ion pressure, which is something like this, and then sweat rate is measured by ventilated capsule method at different press, and sweat lactate is measured by this method. So what we did that we compared the sweat lactate and sweat rate relationship, whether this is linear or non-linear, something like if the lactate threshold exists. So when we do group mean, the sweat lactate content and sweat rate relationships relationship looks like very linear. This is correspond to some previous studies, but the previous study doesn't measure sweat content directly. So what we did actually, we measured sweat content directly and reported this relationship. However, when we look individual response, it is a little bit different. And most participants showed kind of a threshold-like response in increasing sweat lactate content in response to sweat rate. But some are not really, so there are many individual variations. So when we compare the response sweat rate threshold for increasing sweat lactate content between trained and untrained individuals, we found higher response in trained individuals. So this means that there is a possibility that there is a kind of metabolic alternation after exercise training to induce more aerobic metabolism in sweat gland. However, this is a little bit challenging. So here we demonstrated the potential presence of a breakpoint in relationship between sweat rate and cell lactate concentration, at least in several participants. This response is augmented in exercise-trained individuals. However, this technique is still challenging, so I need more development. Okay, this is just a summary I have talked about the methodological and physiological challenges to measure sweat sodium and sweat lactate. And also I will introduce some of our studies. That's all. Thank you very much.
For the excellent presentation. So we open to the questions. So any questions from?
Okay, thank you very much. This is very interesting topic. So my name is Sri Martinez. I come from Indonesia, Semarang State University. I would like to know, this is the new information that you use the lactate sensor for your research. And this equipment you make by yourself or by have the product, new product first?
So I don't make myself, but my colleagues, I work with another university in Japan, also another one company in Japan to make this product. But I don't think they sell now yet. They want to sell, but not yet.
Okay. And the second is about sweat concentration, and that I saw in your graph that is have a sweat concentration that is have I think salty and well concentration. So what does it mean that is salty? It mean the natrium is higher, or this is the indicator that for the sweat that you do. What is the indicator? The measure of sweating. And I saw also in you use the equipment to make induce sweat.
You are talking about
Yeah.
which one? Difficult. Sorry, I'm not sure what you are talking about. We can do break. I'm sorry, what? Okay. So, what do I mean highest sweat sodium concentration? So, yeah, I think this is good to see the to interpret sweat sodium concentration. So average value is like 40 millimole per liter. Yeah, average 40 to 50, although it can differ depending on the skin lesion. And then this is average. So I think higher value can be 60 or 70 millimole per liter, and lower value can be 10 to 20 millimole per liter.
Thank you for a good presentation. I'm Sang-Lee from Western Michigan University. Actually, this is not my field, but it was a very good presentation. I just want to make one comment on your slide. Can you go back to those number 19? So I really agree with your opinion. Interpretation is very important, right? When we look at the data, so unless we interpret the data, it's kind of very risky. But especially on the left one, okay? From my understanding, you highlighted that. Yeah, that's good, right? So, for example, we have the line grid, right? Can have some different variability across the data. That's why we have to be very careful interpret the data. That's good. But I also add one more comment on this way. For example, these two lines are the kind of the similar pattern, right? That means in order to look at some effects of some I don't know the variable that, especially when you have two different patterns, we have two different starting or baseline data. Yeah, that's why we have to take a look at that point. For example, we have to use the variable like covariate, okay, to remove the errors coming from potentially two different sets of data in terms of the baseline, the value. So I want to just make a comment on that. You have to be careful of, interpret those kind of data, especially when you have two similar patterns. Okay, by adding the baseline data, it's like covariate. But relation should be good, but to look at some effects of sweat value, whatever it is, on the value, you have to look at the covariate.
Your comment is, sorry, I'm very understand what you mean.
Especially when you have two similar patterns, which have two different baseline data.
Yeah. Okay.
Yeah.
You mean we should consider about the baseline?
Yeah.
Okay. So that's a good point. But actually, we don't have baseline because baseline of sweat rate is 0 before exercise.
I mean, the first data is one. Right?
Yeah, but I think if we can produce the same or method, that's okay. But when we exercise, sweat rate can differ depending on the date or before and after heat accumulation. So it means that the initial data also can differ.
Yeah.
Depending on the individual. So, maybe good to make a kind of standardized point to interpret the data.
Thanks for the question. So any questions?
Hi, my name is Chang Hai Wang. I'm from NCKU. Thank you for your presentation. Very good talk. And my question is, I'm very interested about measuring the lactate in our sweat. So have you ever compared the sweat lactate to the blood lactate? Because the blood lactate is always the golden standard for us.
Thank you for the question. So we did just a pilot testing, but I know that several studies try to measure sweat lactate as an indicator of blood lactate concentration. But this is physiologically tricky because sweat lactate is mostly came from sweat gland cells. So it can come from the blood, but concentration is quite high from the sweat gland itself. So, for example, what we did in our lab is that when we do passive heating, no exercise, but participant sweat and the blood lactate don't change a lot, but sweat lactate concentration increases a lot depending on sweat rate. So, in my opinion, sweat lactate concentration doesn't reflect blood lactate level.
Due to the time limit, so we need to close this section. If you have further questions, you can ask Dr. Amano later. Okay, so thanks again for your very comprehensive presentation.
再次掌声感谢为我们带来精彩的演讲。Please give applause again. Thanks for giving us an excellent presentation. 请各位与会人员注意，中午便当请至本系馆1楼领取，并可在1楼92177与92185教室用餐。本会员监事请移动至1楼92171教室用餐，并召开以监事会议。另外，提醒本会员年度大会将于12点50分在繁城讲堂召开，请所有会员们请务必出席参与。